Hi. Hi, this video is about lac operon. Lac operon is an important topic in molecular biology and is also asked in various entrance examination. Come, let us see about lac operon. So, what is an operon? An operon is a functional part of a gene that has many structural genes under a single promoter. For example, you can see lac operon, tryptophan operon, etc. Here we are seeing about lac operon. Here we have lac Z, lac Y, and lac A. These are structural genes. There are more than one structural genes, but they are encoded by a single promoter on a single unit of operon. Here in lac operon, it is present in E. coli. This E. coli or Escherichia coli bacterium uses either glucose as lactose as its energy source. But glucose is always the first choice. When both glucose and lactose are available, it always chooses glucose. So you see, when glucose is used as a source, lac operon is switched off. When glucose is not present, then E. coli uses lactose. When lactose is used as a carbon source, lac operon is on. So when glucose is present, lac operon is off. Whereas when glucose is absent and E. coli uses lactose, lac operon. So why when lactose is present only lac operon is on? Because as you see, lac Z, lac Y, lac A codes for what enzyme? Lactose metabolizing enzyme. You can see lac Z codes for beta galactosidase, which breaks down lactose into glucose and galactose. Lac Y is a permease. Permease means creating pores in the cell so that lactose can enter into the cell of E. coli. Lac A is transacetylase. So what are these? These are lactose metabolizing enzyme. If the lactose is present in the uh, E. coli as an energy source, we need enzymes to metabolize to lactose. So whenever lactose is present, E. coli switches on its lac operon. Whenever glucose is present, E. coli does not switch on. It switches off the lac operon. So you can see who controls the switch on and off. That is regulatory gene. These three are structural genes. Apart from this, LAC-I is a regulatory gene. This encodes for a protein called repressor. So, what is the action of repressor? Now we can see about it. Okay. When the glucose is absent, repressor is active. Repressor binds to an operator, which blocks transcription. To understand this, we can look at a picture. If you can see in this picture, in the absence of lactose, no lactose, only glucose is there. So no lactose is there. Now, this regulatory gene, LAC-I, encodes for the protein repressor. This repressor becomes active. This repressor binds to operator. This now transcription is stopped. There is no initiation of transcription, no production of metabolizing enzymes of lactose. So, whenever the lactose is absent, repressor binds to operator, no transcription of genes. Now, opposite scenario, there is presence of lactose. Now, this repressor becomes inactive. How? We have an inducer called allolactose. This binds to repressor, makes it inactive. Inactive repressor cannot bind to operator. If repressor does not bind with operator, nothing can happen to the transcription. Transcription takes place smoothly. So all the enzymes for metabolism of lactose are produced when there is presence of lactose. So I think you understood what is operator, what is a repressor, how the repressor binds to operator in the presence of in the absence of lactose, how the repressor does not bind to operator in the presence of lactose. Now, coming to the next concept, catabolite repressor protein. So, this catabolite repressor protein, or also called catabolite activator protein, this is activated by another molecule called CAMP. So, how CAMP is produced? When glucose is present, CAMP levels are low. So, when CAMP levels are low, it inactivates CRP. The opposite scenario, glucose is absent, CAMP is high, CRP is active. This CRP or CAP binds upstream of promoter in the RNA polymerase, where in the carboxy terminal domain of RNA polymerase, and increases the expression of the genes. So whenever RNA polymerase, what's the function of RNA polymerase? It initiates a transcription. So more transcription of structural genes. Okay. Now this table provides a summary. Whenever both glucose and lactose are present, lac operon is off. There is a basal level of transcription. What is the basal level of transcription? We studied there is no but there is always a basal level of transcription. Basal level of transcription means there is always a minimum level of transcription of the enzymes. You see, LAC-Y encodes for an enzyme called permease. What is the function of permease? It allows lactose to enter into the E. coli. Now, consider if there is no permease, how does the E. coli know whether there is lactose is present or absent if it is not able to enter in the cell? Therefore, always there is a basal level of transcription of these enzymes so that some permease is present, some lactose would enter the cell. it can be sensed by the E. coli, and it can now the regulatory gene can be on or off. So lack operon can be switched on or off accordingly. Now, these are some extra points. Operator, there is a two types of operator: primary operator and secondary operator. Primary operator is where usually repressor binds. Secondary operator has some less affinity. Apart from that, you also have to remember that the LACI gene encodes for repressor. The repressor is a homotetramer. It consists of four subunits. And you can see the allolactose binds to the repressor also. So coming back to this table, we saw that this basal level of transcription is required always. So whenever there is a lacoperon is off, also there is a basal level of transcription. So when will be the lacoperon off? When both glucose and lactose are present. Even when both glucose and lactose are absent, also lacoperon is off. So when will it be on then? When lactose is utilized as a carbon energy. So when glucose is absent, then only E. coli uses lactose. Then lacoperon is on, then there is activity level of transcription. Now consider this scenario. There is no lactose, only glucose. If there is no lactose means who needs lacoperon? So lacoperon is off. So in this scenario, when only glucose is present and no lactose, lacoperon is off. That means repressor bounds to operate. That means no transcription. So I think this video provided a very good understanding of lacoperon. For further information or further detailed mechanisms of lacoperon, like how heterozygotic conditions can affect the expression of genes and enzymes, how the transcription is activated, we will meet in the upcoming videos. If you like this video and you thought that something you understand from this video, please like and share this video. Thank you.